Start. Hello everyone, welcome to your first class of Kalam batch. So do write in the chat box if you are able to hear me and if the video is clear. Good evening. Okay, so am I audible? Okay, great. So without further delay, let's start today's class. So today is your first class of biology, right? And the topic is cell, the unit of life. Okay. So before we start with the lecture, I will tell you few basic things, okay, that you should keep in mind while attending my classes and referring to my notes and all the things. So the first thing is that when the class is like you are watching the class live, then your only focus should be on what I am saying and what I am writing on the board, right? If you are able to match your frequency of making notes with my lecture, then you can make the notes then also with the class also. But if not, then don't make the notes. You will get the all the material, okay? And you can further on you can add it to your notebook, right? So this is one thing. Another thing is that how will we start the class? So basically in my class, what we will do is, like, for every topic, right, we will be covering, first thing is NCERT lines, right? So as you know, you are also preparing for NEET exam, right? In NEET, NCERT has the most of the weightage for biology and most of the questions are directly from the lines of NCERT. So what we will do is we will first understand the topic, then we will refer to those lines so that when you read NCERT yourself, you have the confidence that yes, I am able to understand this particular topic, right? So this is the first thing. Second thing is, in my class what we will do is we will cover the PYQs, okay, from that particular topic that I am teaching. Mainly we will cover the PYQs of IIT, NEST as well as NEET, right, and all other exams which are applicable. So the PYQ practice will also happen within the class itself, right, so that when you yourself practice questions you don't have any difficulty. And apart from that, what we will do is we will understand everything in detail, right, each and every process, each and every topic, and we will link it to different let's say for a topic you have to understand something from physics or something from chemistry, then we will do that so that you have a holistic learning, right? So this is a interdisciplinary class, not a proper biology class, but this is interdisciplinary, right? So these are few things which we will be doing. And now talking about to the people, so in this batch you have people who have only PCB, who have PCM or PCMB students, right? So we have all the different varieties of people. So for the PCM guys, you should not take bio for granted, right? If you think that you can learn and the NCERT and you can just rote learn it and then you can solve all the questions of IIT and NEST, then you are in some dreams, because that is not true and that will never happen. You have to understand biology, and then a few things which are there that you have to learn, that I will tell, right? And apart from that, the PCB students who have a good command over bio, right? So for you, you know what to study, right, for NEET and all. But the thing is, we will cover IIT and NEST also, and IIT and NEST questions are much more difficult as compared to your NEET questions, because NEET is similarly like anything that is directly picked up from the NCERT, but for IIT and NEST you need thinking, you need a thought process. So we will develop that. As soon as you attend all my lectures, you will be able to do that, okay? So the before starting the class, I want to say this, that if you are droppers and if you have like taken a drop, so you have to work hard, right? You have to focus on whatever is being taught, and yeah, I guarantee you that you will score the maximum in biology because you have attended my lectures. So that is my promise. Yeah, nest is numerical base. So in today's class also you will see from this topic only in cell biology how many numericals and other things can be framed, right? So now, so in today's class first we will understand what is cell, right? What is a cell? The next thing that we will be understanding is that what is the difference between prokaryotic cells versus eukaryotic cells. Apart from that, apart from this difference, we will also look into few techniques, for example microscopy. So if you know in next exam from this particular topic cell, the basic unit of life, you have the microscopy, right? So someone is saying, so for study materials is NCERT enough or any other book will you suggest? So NCERT plus my class notes plus my lecture, that is enough, and the material that you get in the batch, that is your questions for practice, that along with the PYQs, that is enough. Okay. So yeah, so now we will cover microscopy, we will understand about hemocytometer, okay, and in between this few small topics, few important things will also come that we will discuss. So let's start the class. So the first thing that you need to understand it, what defines a cell, right? What do you understand by the term cell? Can you guys comment down? What do you understand by the term cell? Basic unit of life. Yes. Any other improvisation that you can do with this definition? Fundamental unit of life is cell, unit of life, okay, basic unit of life, structural and functional unit of life, very good Amrita, okay. So yes, so if you understand in layman terms, right, in layman terms if you understand, cell is nothing but a compartment, right, and it is the compartment that was required for life to sustain. Why I am saying so? So during the time of evolution, during the course when all our you can say when everything was setting up in this world, what happened was for life to sustain you require a compartment, right, which separates. So let's say the outer environment, it is made up of water, right, H2O, and if something has to sustain, right, it has to be stored, right, it has to be present inside this boundary, okay, and for that to happen this boundary it should be hydrophobic, right, this boundary should be hydrophobic. What do you understand by the term hydrophobic? Can you guys tell? What do you understand by the term hydrophobic? Repel water, water hating, yeah, correct. So because of this water cannot enter inside, right, so this boundary should be hydrophobic, and all the things which are required for life to sustain, whether it is a genetic material, whether it is your DNA, right, DNA, RNA and proteins, right, all these things they will be present inside this boundary, right, and what they will do inside the boundary, so the process of protein synthesis will happen, right, and you know everything life, if you say if you want to summarize life, life is a manifestation of proteins, right. So if you want to understand it in simple terms, if you have watched a movie, right, so in that case what you can understand is DNA. So RNA is made from DNA and then proteins are made from the RNA. So if you understand DNA, DNA can be said as the director of life, right, which has all the information for life to sustain, to function, correct. But if you talk about RNA, RNA you can say as a producer of life, right, you imagine a movie, so you have director, producers and actors. So similarly DNA can be referred to as the director of life, RNA as producer of life, proteins, they are the actors, they are something that are carrying out those particular functions of life, right. So this was a very layman introduction to cell. This was the main idea why this compartment we are referring to as cell, and this was very correctly said by one of the student that cell, we can say it as the fundamental and structural unit of life. Right, fundamental and structural unit of life. So now what is the meaning of this statement? Many people know about this statement but they don't understand what is the actual meaning. Fundamental means anything which is smaller than this unit cannot survive, right? Because fundamental means it is the building block, correct? So this is the building block. Yeah. Yeah, so fundamental means it is the building block, correct? So that means that anything which is smaller than this, for example, if I ask you a question that is, what is living? Okay, and I have few options. I have ribosome, I have Golgi body, I have nucleus, and I have cell. So can you tell me in the chat what will be the answer? What is living? Ribosome, Golgi, nucleus or cell? Okay, the slide size, yes. Yeah, I hope now it is fine. Yeah, so what is living? Ribosome, Golgi, nucleus or cell? Yeah, cell is living. So why cell is living? Because cell is the fundamental and structural unit of life. Structural unit of life means it is able to it has a proper structure, a compartment, and that also perform functional. So it is a structural and functional unit of life also, right? So you can also add functional here, correct? So anything which has to be called as living, it should be able to understand, like anything which has to be living, that should have a proper structure, it should be able to perform function, and also it is should be the you can say it should be made up of these building blocks called as the cells. Now if you understand these lines of NCERT that say what is a cell, correct? So unicellular organisms are capable of independent existence, correct? And performing the essential functions of life, right? So anything less than a complete structure of a cell does not ensure independent living. That is something that is being referred in the NCERT, that anything which is smaller, right? Is the screen not clear? I hope it is readable, right? Yeah. So anything which is smaller, right, that is smaller than this fundamental unit of life is not considered as living, right? So the first thing is that the cell, right, it should be of independent existence, right? It should be able to exist independently, correct? Right. So that is what is being referred to in the referred in the NCERT. Now we talk about few scientists. So if we start with this, before starting to that, we can talk about one thing. So you remember I talked about that compartment, right? I talked that this compartment, this is hydrophobic, and since this compartment is hydrophobic, it is able to form a secure environment for the inside materials in this water surrounding, right? Because water is polar, right? It is hydrophilic. So this water, it is being repelled by this, and basically this is a very secure environment for all your DNA, RNA and proteins. Now we have to understand this boundary, right? We need to understand this boundary. So this boundary, it is made up of lipids, okay? This boundary is made up of lipids. We will understand lipids in detail, but for now I will explain you like how this whole boundary is formed and a small misconception that people have regarding lipids. So, what happens is mainly you have three kinds of structures. Okay. Okay. So, if we understand the first structure, if you talk about, so let's say you have this hydrophobic head, sorry, the hydrophilic head and hydrophobic tail, that is this chain. This tail is made up of fatty acids. You know what are fatty acids? They are long chain carboxylic acids. So this is the hydrophobic part, and this head, this is hydrophilic, correct? So this part is hydrophobic, this part head is hydrophilic. Now overall this structure, if you see, this structure is basically like a triangular shape or a conical shape if you visualize in 3D. So we call this as asymmetrical, right? We call this as asymmetrical. Now what happens is when you are having these kinds of lipids, you form a structure where the hydrophilic part points inwards and the hydrophobic part points towards the outer side, correct? So these kind of structures which are formed, they basically they are called as micelle. So this is micelle, right? And now if we talk about the different structure that exist, that is basically your lipid bilayer. So for that structure to exist, you have two chains of the long chain carboxylic acid, and if you visualize this in a particular shape, this structure will look like a cylinder, right? And we call this as a symmetric structure. This is a symmetrical structure. So now when this symmetrical structure, it is put in water, okay? Let's say you have a beaker of water, you put the structure there, right? And now what will happen is this will form something that you can call as basically you have this lipid bilayer, right? So the cell membrane that you are having, that is made up of this lipid bilayer, where these kind of structures they are present. And if you think, what happens is this is the lipid bilayer of the cell, right, where this part is hydrophobic and this part is hydrophilic. So this is something which we call as a lipid bilayer. So this is how the lipid bilayer is formed, and this lipid bilayer is present as your cell membrane. That is the membrane of that compartment that we saw, right? So that is how basically all these things happen. And in solutions, as you know, this hydrophobic part will be pointing towards the you can say inside, and the hydrophilic part will be pointing towards the you can say outside region, so that it can interact with water. But that is one thing. The another thing is, so here as you saw this thing, so if you are putting this this whole micelle kind of thing, right? So here it will not be interacting with the water. This is the case with the organic solvents, right? If you have any organic solvent which is also non-polar, then this part can interact there and the aqueous environment, this will survive. But if you are putting it in some aqueous environment, right, so then what will happen is, then this is the case. Right. So now the hydrophilic part will be outwards, this is the water, and the hydrophobic part will be in the inside the core, right. When will they become symmetrical? Okay, so the symmetry comes from the structure itself, right. So these structures which have a two fatty acid tail, right, so this is causing the symmetry. You imagine like so you imagine a person, right. So this is the person. Now if the head of the person is too large, correct, and if the person body, if you imagine this body, right, if my body is like very thin and my head is too large, then I will become asymmetric, right. I will be you can visualize me as a conical kind of structure, right. But if you are having a person whose body is not that much thin, it is in proportionate with the head, so then it will be called as a symmetrical structure. Yeah, yeah, I am talking about the lipids, basically what kind of structures they can form. And yeah, so that is how the lipid bilayer is formed. And then if you talk about another structure, that is so basically with the symmetrical ones only, yeah, you can have another structure which can form, that is called as liposomes. Under certain conditions, these liposomes, they are, how do they look? So basically what happens is, this bilayer only, this lipid bilayer, if it folds, right, you imagine this to form as a sphere, correct. So if it forms as a sphere, then what will happen is your mainly so inside what will happen, inside you will have this kind of structure, correct, and here in this particular case you will have this kind of structure. So basically this is the bilayer which is folded, and when it folds it forms something like this, and overall if you pack this structure, right, yeah, so overall if you pack this particular structure, you get something called as a liposome, in which this environment, okay, if I mark it with white color, this environment, basically here you have the aqueous environment. So in the aqueous environment what will happen is, okay, how the symmetrical lipids form the bilayer, yeah, so you want to understand that. So see, what I am telling is, if you understand the first part, right, this part, so you see this is asymmetrical, the head is so large, the body is so thin, correct, yeah, the body is so thin, the head is so large. So in this case the structure is asymmetrical, right, and the one thing that is there is you have a single tail, correct. So if you have a single tail, then what happens is the head is here and the tail is like this. So this structure we are calling as the micelle, that forms for your detergents and all, right. And the next thing is, now you are talking about the symmetrical lipids, right. So let's say the head is this much and the body is also proportion with the head, like you have the two tails, that is making this structure as symmetrical structure, correct. So this symmetrical structure, if you understand this, right, the bilayer, lipid bilayer kind of thing. So in aqueous solution what happens is, the head part, since it is a bilayer, right, you have two of these molecules, this lipid molecules, the head and the tail, which are oriented in this manner that they form a bilayer, because here you have the first layer and here you have the second layer, right, and this is what is forming your cell membrane. Now comes the third structure, that is your liposomes, correct. So what happens is, if the bilayer, if you have this kind of structure where first layer that you are having, okay, so this whole you can call as liposomes, you can call the structure as your vesicle also, right. So you have heard about vesicles, right, those structures which are used to transport molecules from inside and outside of the cell. Okay, so see, when your this molecule, this it is put in water, right. So what happens is, when you are when the head is hydrophilic and it is in water, what will happen is, this head, this is hydrophilic, correct. So the head, it will be oriented in this manner, correct. So that no, not in this manner, the head, it will be oriented outside and the hydrophobic part will come to the inside, right. So that is how overall the structure is formed, okay. So okay, so is the liposome hollow? No, the liposome is not hollow. Inside you have the aqueous environment. You have water where all your DNA, RNA and all the materials can be present. Okay, in this aqueous environment you can, in this aqueous environment you can have okay, yeah. So in the aqueous environment you can have your DNA, RNA, right, the genetic material as well as your proteins. So these are used as delivery vehicles. Okay, these are used as delivery vehicles. So what do you mean by the, yeah, so in case of Abhishek, in case of liposome, what happens is your head is in this manner and the another layer that is there that forms a spherical kind of a, you can say, particle kind of thing, right. Inside this particle you can have, in the inner environment, in this aqueous environment you can have either your DNA, RNA or protein, and that is used as a delivery vehicle to deliver inside the cells any of your DNA, RNA or protein, because see, this is the lipid, right. So it will fuse with the cell membrane and the inside material will come inside the cell. So that is the part. Yeah, okay. Can I say that liposomes have thick surface which is hydrophobic? Thick surface, you can't define thick or thin, but the thing is that when the lipid bilayer, it can form a structure called as vesicle, in which inside you have these hydrophilic groups and outside of this part has the hydrophobic groups, and finally inside it is the hydrophilic groups. So here if it is water, then these groups can interact with water because they are hydrophilic, and inside also you can maintain aqueous environment, and in between you have this hydrophobic environment. So that is how it is used as a vesicle. Okay, why it is used for delivery vehicles? So if I tell you about myself, so I have worked in the field of vaccines, right. So there is something called as mRNA vaccines, in which what happens is that let's say you have a mRNA, okay. So mRNA encoding any protein. So if you put that mRNA inside this vehicle, okay, inside this environment, then you put that mRNA in, you inject it in your body, it will go through your blood, it will just be, it will just go to your cells, and because this is lipids, right, so it can easily fuse with your cells and it can deliver the inside content, okay, inside the cells. And once the mRNA is delivered, it will form proteins, right. MRNA forms proteins, and those proteins, they are displayed on the surface of the cell like this. And I will teach you, I will help you in immunology to understand that this cell surface expression of proteins, it is helpful in generating immunity, immune response. Your immune cells will recognize this foreign protein, they will make the antibodies and all. So that is the idea, okay. So that is why the delivery vehicles are used, okay. Yeah, liposomes are artificially made. So there are different methods of making the liposomes, okay. And one is like you can just rapidly pipette mix the lipid and the DNA solution and in some ratio, and then you can form these spherical structures, okay. Yeah, how does it fuse with the cell membrane? See, this is made of lipids, this is the head group. In your cell membrane also what happens is you have this kind of structure, right. So if a liposome is coming, it can easily fuse, this is hydrophilic only, and it can pass through, right, it can be delivered. Is that clear? Because there is no difference in the charges, because both are hydrophilic. It is not like one in the outward side is hydrophobic. So since the polarity is same, they can fuse inside the cells and it can deliver your content. Is this part clear to everyone? Any doubt? I told liposomes, they are used for delivering genetic material, DNA, RNA, proteins inside the cell, so that you can have different functions in cancer immunotherapy, in vaccine design. So the idea is, if you deliver, let's say, mRNA, okay, so the mRNA, it will be made into protein, and that protein, let's say you are making the protein such that it can be displayed on the cell surface, then immune cells can recognize foreign protein, they can make antibodies, and that's how it works. So I will tell everything in detail, but the main purpose is therapeutics. These delivery vehicles are used as therapeutics, Okay, is this part clear? For making different drugs. So this is how mRNA vaccines work. You make this lipid nanoparticles and you put the your genetic material inside those particles so that you can deliver it. I will teach everything in detail, but this was an introduction to what different structures these lipids can form because of symmetry and asymmetry, and we will cover this in detail also in the next class where we will see about plasma membrane. Okay, today I am just giving you an intro. Okay, yeah. So now let's start another important topic, that is about, so if what happens is, if cells have to survive, okay, so they want to take a particular shape. The compartment, you can ask me why the compartments are only spherical in shape, right? There can be different, yeah, liposomes are mostly artificially made. Yeah, liposomes are made artificially. These are the vesicles. There are different versions of these, okay, that I am not going into great details. I will teach when I will teach immunology, but yeah, they are made artificially, and how they are made I will also cover that. But for this part I want to tell you the difference between lipid bilayer, micelles and liposomes. Is that clear to everyone? John is saying how they are used as delivery vehicles. Okay, John, see, let's say you are here, right? And this is the particle, the liposome, the vesicle that I am talking about, correct? Inside this vesicle, what is there is either your DNA or let's say any of the material that you want to deliver, right? And what I did is I just injected it inside your body. Inside your body, what will happen is, let's say you are having this cell, right? This is your body cell, and this vesicle, it can fuse because it is also made up of the same groups, it is also lipids, right? So it can fuse with this membrane and it can deliver the inside content, that is your genetic material inside, let's say RNA. It delivered RNA, now your cell will make protein from it, right? And that is how, if your protein is made, then what will happen is, if my protein has a transmembrane domain, that it can come in an anchor here, right, on the cell surface. This is your protein. Now comes the immune cell, T cell, B cell, all immune cells, they recognize the protein and you get immune response. That is how these things work. Okay, yeah, it is similar to basically you can't call it as yeah, it is similar to endocytosis. Yeah, correct, Ritika. So that is the idea how it works, okay, and why it is made and like that. Is it clear? Anyone has any other doubt in this part? Then we can start the next thing. Is this clear to everyone? Okay, Hiral. Okay, no doubts. Okay, great. So the next topic is, if we talk about the basically, so for what happens is the cell can take any shape. It could have been like a rectangular or a cuboidal kind of thing, right? It could have been this kind of shape. It could be this kind of shape, right? There can be a different shapes that the cell could have taken. But the most of the cells, if you see, they are these spherical cells, cells, right? They are spherical. So why sphere? So why the cells are spherical? Can you tell? Yeah. Why the cells attain a spherical shape? To decrease surface area, because the ratio of volume and surface area is high, least surface area. Okay, okay. Minimum energy configuration. Yeah. For any if you say to occupy the maximum volume, right, and minimum surface area, for that particular purpose sphere is the best geometrical figure that you can have. And mainly so if we talk about the area of the sphere, right, so if we talk about the area of the sphere, so area of the sphere is 4 pi r square. This is how we define the area of the sphere, right? This formula everyone knows, correct? And the volume of the sphere, if you define, that is 4 by 3 pi r cube. Volume of the sphere is 4 by 3 pi r cube. This is also clear. Everyone knows these two formulas for sphere, correct? So now if you calculate the surface area to volume ratio, this is surface area to volume ratio. If you divide all this, right, you will get simply 3 upon r, correct? You will get 3 upon r. Can you do this calculation? 4 pi r square divided by 4 pi r cube by 3. 3 goes upwards, this cancels out, you get 3 upon r. Is this clear? Clear? So it means surface area by volume is proportional to 1 by r. That is how the surface area to volume ratio is dependent on the radius of this particular sphere, that is your cell, correct? Now what the cell wants, right? What the cell actually wants. So if you talk about the cell it wants to have maximum volume, right? It wants to have the maximum volume so that it can carry out all its function, it can have all its you can say the activities which are happening inside the cell. Okay, Pawan, do you have any doubt? You can ask in the chat. Okay, so the cell wants to maximize the volume, correct? So if we talk about the surface area to volume ratio, right? So what happens is in this particular concept, if we talk about, so for a particular cell, what is happening is this is the surface area to volume ratio that is dependent on 1 by r, right? So if this ratio is high, right, if surface area to volume ratio, this ratio is high, right, that means that the surface area is high and the volume is low, right? That is the case. So this is one possibility. Another possibility is surface area to volume ratio is low, right? So the surface area is low and the volume is high. So there are two possibilities for the cell. Either it can take its surface area to volume ratio as high or low. So what will the cell prefer? Does it want to have high ratio or low ratio? Can you tell? No, I am not saying does it want to have. I am saying there are two options. Let's say you are cell. So you have two options. Either you can have high surface area or low surface area to volume ratio, right? So you have these two options. So which option will you prefer? That is what I am saying. Yeah. So many people are saying low ratio. Some people are saying high ratio. Okay. Okay. Option 2. Okay. Okay. So let's before answering this, let's solve a question. Okay, so this is NEST 2022 question, right? And you can read the question. Okay, you have two spherical cells P and Q with radii of 1 micrometer and 0.01 millimeter. Okay, choose the statement that is correct regarding these cells. So you have four statements. So can you answer? Okay, I will tell that particular part. Before that, you just solve this question. Okay, Abhishek? Yeah. So in this case, you have two cells, right? So one cell, if we write here, you have P and Q. If we talk about the radius, correct? So for the P, you have radius as 1 micrometers, right? And for Q, you have the radius as 0.01 millimeters, right? Now you have to tell that which has a more surface area or volume or these kind of things. So how can you do these kind of questions? Can you tell? Okay, people are saying option number B. Okay, fine. So if you see this, right, first we have to convert everything into same units, right? So it's better if we convert this particular thing, right? So what happens is 0.01 mm, okay? So if we write this, we can simply say this is 10 to the power minus 2 millimeters, right? And you know 1 meter is 100 centimeters, that is 1000 mm. So if we talk about 1 mm, that will be 10 to the power minus 3 meters. And 1 meter is what? 1 meters is 10 to the power 6 micrometers, because meter is a bigger quantity. So here you have 10 to the power 6, right? So here you get to know that this is 10 micrometers. So this is the first thing, that this cell is 10 times larger as compared to the Q is 10 times larger to P. That is something you understood, right? So this is 10 micrometers, correct? Now the P surface area to volume ratio is 10 times greater than that of cell Q, right? So this is the option. So if you see the surface area to volume ratio, as I told you, if we calculate this ratio, right, for P, so that is dependent on 1 by R, that is 1 micrometer, and if we calculate the ratio for Q, that will be dependent on your 10 micrometers, right? So with these, what you understand is that the surface area of cell P surface area to volume ratio is 10 times greater than that of Q. That is something which we understood. So option number B is correct. So all the people who have answered as option number B, they are right. And if you see this thing, cell P has a surface area 10 times greater than cell Q, right? So if you see the only the surface area, then this is not 10 times, because surface area is something that is dependent on R square. So if this is 10 times, this has 10 times more radius as compared to P, then the surface area will be higher by the factor of 100 and not 10. So this is a wrong option. The volume of Q is 100 times larger than cell P, okay? So the volume is dependent on R cube. So that will be by a factor of 1000 and not this, right? So this is also wrong. And if the radius of P and Q is double, then the volume and surface area is also double. No, this is also wrong, because the dependency is wrong, correct? So is this question clear to everyone? Any doubt? Any doubt in this question? Okay. It is clear, okay. Now this thing, okay, this thing is your homework. We will cover it in lecture 2, okay, when we will start with the topic of cell membrane, right? There we will discuss this, we will understand the structure of the lipids, the cell membrane in detail and everything. So this is the homework for lecture 2. You have to understand this. And yeah, anyone has any doubt, they can write in the comment. Yeah, so this we will cover in the next lecture, okay? So do attend that. And moving forward, then we can start with this, I guess, okay. In this question, does anyone have any doubt? It is clear, right? So basically the question says you have two cells, one is cell P, another is cell Q. The radius of the first cell is 1 micrometers, the radius of the second cell you have is 10 micrometers. So you can simply say that the radius of cell Q is double as that of cell 1. And if you talk about the surface area to volume ratio, then that is dependent on 1 by R that we saw. So for Q it is dependent on 1 by 10, for P it is dependent on 1, that is 1 by 1 micrometers. So if you divide these two now, if you are not able to visualize, then the surface area to volume ratio of P to Q, right? So if you divide this, it will be nothing but 1 by 1 divided by 1 by 10, that will be 10 by 1. So you can say that if this is P by Q, then the surface area to volume ratio of P is 10 times as that of Q. So I hope that is clear, okay. right. So now we come over to the scientists who are related to cell biology, who have done the major discoveries. So the first one is Robert Hooke. I hope you know the name. So this part is important for NEET. This part is important for NEET. Okay. In IIT there is a very less probability that these questions can come. This is important for NEET only. Okay. So in the year 1665, okay, Robert Hooke observed thin slices of cork under a microscope. Okay. So cork, you know what is cork, right? That is, you know about the bark of the tree, right? So from that only, in the anatomy I will tell you what is cork. So from the cork he observed basically these tiny box like compartments like this, and he called these compartments as cellulae, that is cells. Okay. So basically these cellulae or cells that he observed, these were dead. Okay, because the cells of the cork, they are only having these cell walls and they are dead cells. Okay, so he observed the first dead cell, not the living cell. Is this important for IIT? For IIT this is less important. Okay, this is important for NEET. NEET, in the re-NEET also you have a questions from scientist name. So yeah, this is important from NEET perspective. Okay. So yeah, so he observed the first dead cell. Then comes Antony von Leeuwenhoek in the year 1674. He was the first scientist to observe and describe living cells using his self-made microscope. Okay, and what is a microscope we will cover today. He observed bacteria, protozoa, sperm cells and other microorganisms, calling them animalcules. Okay, so he called them animalcules, and basically he was the first scientist who observed the first living cells. Okay, so this was the first guy who observed the first living cell. First living cell. Robert Hooke observed first dead cell. Okay, is this clear? If you want zoom, then I can do. Okay, I hope this is clear now. Then comes Robert Brown in the year 1831. He discovered the nucleus, okay, in the plant cell. And then comes Skeleton. So he was a German botanist. This is important to know. He was a botanist. In the year 1838, he concluded that all plants are composed of cells and cells form the tissue of plants. So he said that all the plants that you are able to see today, they are composed of these cells, okay, which are the fundamental structural and functional unit of life, and they form the tissue of the plants. That is the plant tissue, whether it is xylem, phloem, any of the plant tissue, vascular tissue, they are being formed with these cells. Then comes Swan. So he was a British zoologist, okay, and he studied different types of animal cells, and he concluded that all animals are composed of cells. He distinguished between the cell wall plants and animals. So in case of plants, I hope you know that you have a cell wall in addition to the cell membrane. What is the cell wall, what are its components, everything we will discuss in the next lecture. But the main difference between plant and animal cell, you can say, is the difference in the outer structure. The first difference that you can see is the cell wall, correct? That is present in plant cell, absent in animal cells. So in the year 1839, Swan, he formulated the same thing for the animal cells, Skeleton said for plant cells, and together Skeleton and Swan in this year, they propose the cell theory. So now what does the cell theory says, correct? So we need to understand this. So according to the cell theory, all living organisms are composed of cells. All living organisms, they are composed of cells. So if you are living, you are made up of cells. Okay, yeah, the Germans were really into cell biology, correct? And the cell is the basic structural and functional unit of life. So this is the statement that we discussed, correct? Now there were some drawbacks of the cell theory, correct? There were some drawbacks. So there were some drawbacks of the cell theory. So can you tell any one of the drawback that you have understood till now? That what does the cell theory doesn't explain? It explains a lot of things. It says that all living organisms. So if you define a living organism is something who has consciousness, who can respond to the external stimuli, right? So all living organisms, they are composed of cells. That is something they said. And every cell, basically you can say that cell is the fundamental structural and functional unit of life. Where do cells come from? Yeah, where cells come from, correct? So the origin of cells was not explained by the cell theory. Okay. The second thing, can anyone tell any other point apart from this? Correct. Can you tell about any other point? So they don't talk about viruses, right? Virions and prions, correct? All these different organisms that you can see, they don't talk about this. So prions are nothing but they are proteins, okay? So let's not go there. But if you talk about the viruses and virions, so basically these viruses, they are the connecting link between living and non-living. Okay, so they are the connecting link between living and non-living. So what are viruses and all those things, these were not mentioned in the cell theory. So this was another drawback of the cell theory. And what are viruses, what are virions, what are prions, we will cover it in biological classification chapter, that we will discuss. Okay, they didn't talk about organelles. Yeah, you can say that they didn't talk about organelles, but the thing is, for the cell theory, as a cell we are talking about, we are not going into the molecular level. That is something that you have to understand. Then to solve this problem, there came another guy whose name was Rudolf Virchow. In the year 1855, he added the third postulate to cell theory, okay, omnis cellula e cellulae. That means every cell arises from pre-existing cells, right? So every cell that you see, that is arising from pre-existing cell, and he modified the cell theory and gave it the final shape. So that is something that he did. And yeah, so this is something that happened, that these guys Caledon and Swan, they like formulated the cell theory, but it did not explain how cells were formed. The origin of cells was not clear. To make it clear, to give it a final shape, there came this guy Rudolf Virchow in the year 1855. He said omnis cellula e cellulae, that means new cells arise from pre-existing cells. Okay. Yeah, it was very generalized. They didn't talked about animal cell, plant cell, right? And all the prokaryotes, eukaryotes, nothing was told. Only thing the theory says is if you are living, right, then you are made up of cells, and cells. Now the question comes, what is cell? So they said that cells are the fundamental, the basic, the, you can say, if this is a cell, then nothing smaller than this can exist. Okay, this is the fundamental unit of life. So that is something that they postulated. So this was about the cell theory. So you can read this part from NCERT. I hope this will be clear to you now. Skeleton botanist examined large number of plants and observed that all plants are composed of different kinds of cells, which forms the tissues of the plant. Okay. And then comes Swan in the year 1839, German zoologist, who studied different types of animal cells and reported that cells have a thin outer layer that is today known as the plasma membrane. He also concluded that presence of cell wall is unique character of plant cells. So these were their independent studies because they were botanists and zoologists, but in the cell theory they didn't mention it. Okay. So I hope these clippings should be clear as they are readable. Okay. So on the basis of this, Swan proposed a hypothesis that bodies of animals and plants are composed of cells and products of cells. So they formulated the cell theory, and it did not explain how new cells were formed. Then came Rudolf Virchow, who said the cells arise from pre-existing cells, and he modified the hypothesis to give the cell theory a final shape. So these are the postulates that were said, that all living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. Yeah. So this is something that was said by Rudolf Virchow. Okay. So this was said by this guy, and this was the basically modified cell theory. This was the modified cell theory. So I hope this part is clear. What was Virchow's proof for his assertion, sir? So basically what he observed is, during cell division what happens is, so you have studied this mitosis and all, right? So in the cell division, you know that one cell basically it divides and it forms two cells. So these observations were the proof for his assertions. Okay. Yeah. So he noticed this particular cell division thing, and he said that all the cells are coming from the pre-existing cells. So that is how he arrived at this. Is that clear to everyone? Any doubt till this part? Okay. So now we have a few questions. Okay. So these are NCERT questions. Okay. So the first question is, cell was discovered by. Okay, it's clear. Very good. So the first question is, cell was discovered by. So here we have not talked about the living cell. We have not talked about the non-living cell, right? We have just talked about a simple statement, cell, a simple word, that is cell. So what will be your answer? So many people are saying D, 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 D. Yeah, correct. The answer is option number D, right? Robert Hooke, he discovered the first cell. Now it is non-living, we know that. So if the question was who discovered the first living cell, then you could have marked the Antony von Leeuwenhoek. Okay, that was option B. Clear? Right. Cell theory was modified to its final shape by. Now, these questions are relevant for NEET, not for IIT and NEST. IIT NEST questions I will discuss further. Okay. Cell theory was, okay, everyone is going with option A, someone was with option D, I don't know who is this guy. So that is Virchow. Okay. So Virchow, he modified the cell theory. He told cells come from pre-existing cells. The simplest way to distinguish a prokaryotic cell from a eukaryotic cell is to check for. Now this is a question that I am putting for your knowledge. I hope everyone knows this, right? So with this question, what are the options? Many people are saying A, B, B. Okay. See, here many people, if you think, okay, so the simplest way to distinguish a prokaryotic cell from a eukaryotic cell is to check for. So basically, in both prokaryotic and eukaryotic cells, you have the DNA, you have the protein, you have a membrane, but what you doesn't have is the nucleus. So the answer is option number B. Why people are going with A? See, prokaryotic cell also has a membrane, right? And eukaryotic cell also have a membrane, so that is not an issue. So nucleus is something, because in prokaryotic cell, the major difference, if you see, what happens is you don't have a defined nucleus. The genetic material is lying here and there, okay, DNA and RNA. So that is something, this space then we call as nucleoid. The space we refer to as nucleoid. That is not a true nucleus. But here you have a well-defined nucleus and the genetic material is present inside. So that is the major difference between prokaryotic and eukaryotic cell. Okay. So we will see this prokaryotic, eukaryotic cells, right? And before, so we will cover prokaryotic, eukaryotic cell, plant cell, animal cell in the next class. Now we will discuss something about microscopy. So have you guys ever used a microscope? If you have used, then type yes in the comments. Yeah, you have used a microscope, that is good. So microscope is something that you will use in this research journey very often, right? So you should know about microscope, what are the important components and how does microscope works, that you will study in the ray optics that Nikhil sir will take. He will explain you about microscope, okay, in detail for all the physics part. My work is to make you understand important things so that you can solve the next questions which are there, okay. So first important component is your eyepiece. The second important component that we have to focus on is the objective lens, okay. So if you understand, here you are having two lenses, okay. Here you are having two lenses. So the first lens we talk about is the objective, that is the objective lens, and the second lens is the eyepiece, right? Now before we move into the lenses, we need to understand what does magnification means, correct? So if you consider this as a lens, right, and here if you are having an object, correct, so if the light is falling, the lens, if this is a convex lens, it will converts the light, correct, to a particular point that you call as the focus, and here you will have the light that will be coming, and finally you will get your image, that will be a real inverted image, and that is how basically the image is formed because of the light. Now if you understand this distance, so if this thing, the center of the lens, okay, if I am referring it to as the origin or O, okay, then this distance of the object from the center, that let's say you are considering as DO, and this distance you are considering as DI, okay. So these are the distances, and let's say this height of the object is HO and height of the image is HI. Then if you have to define magnification, that you can simply feel it as HI by HO, okay. That means with respect to the height of the object, how much times you have increased the height of the, how much times you have increased the height of the image, how much times you have magnified it. So I hope you got the feel of magnification. What is magnification? So that means that with respect to the object size, let's say your object here, okay, let's say this was very small, let's say this was a 1 micrometer object, right? Now you have formed an image, and let's say this image is 1 centimeter. So you have magnified the image from 1 micrometer to 1 centimeter. Are you understanding? So this is something that magnification means. And how many times you have magnified the image, based on that you have different lenses in the microscope, okay. So you can have 10x, right? You can have 40x, okay, and also like that you can have different kinds of lenses which have different magnifying powers. Now the another thing is the eyepiece. The eyepiece, which is this top one, this has a fixed magnification of 10x, okay. So this is fixed, this doesn't change for the simple microscope. This is a simple microscope, okay. So for this simple microscope, this eyepiece thing is constant. This objective can have different magnification values like 10x, 40x and like that. Now the overall magnification of the microscope, that you can denote with capital M, that you can write as the product of magnification by the objective lens and the eyepiece, the objective lens and the eyepiece. So that is how you can write the magnification of the simple microscope, that is the magnification that is produced by the objective. Let's say this one, for example, if you consider, let's say M, in this case, if this is 10x, this is 10x, it will be MO into ME, it will be 10 into 10, that will come out to be 100. So this will be your magnification of this microscope, that you can magnify something 100 times, correct. So That is how you understand, this is something that you understand with the term magnification. Now after the magnification thing, what you need to understand is one more concept. Okay, that concept is about limit of resolution, right? So what is limit of resolution? So let's say you are having two objects, correct? So let's say you are having two objects. Okay, so I hope you are able to, okay, so let me draw it bigger, right? Yeah, so I hope this is visible. So limit of resolution is the minimum distance between two objects so that you can perceive them as different objects. Okay, so let's say if the limit of resolution, that is denoted by capital D, right? If this limit of resolution is, let's say let's say it is 1 meter, okay, correct, and this distance is 10 meters, then you can easily you can say you can easily distinguish these two objects, okay, because the minimum distance to see them as different objects, okay, that is 1 meter, but they are 10 meters apart, so you can perceive them as different objects, correct? That is something you should know. Now what happens if this distance, it is very small, right? It is, let's say, 0.5 meters. So if the distance is 0.5 meters, then you will not perceive them as separate objects, you will perceive them as a single object, because in this particular case the limit of resolution was 1 meters, and if two objects, let's say these are 0.5 meters apart, then they will be perceived as a single object. But let's say if they are 10 meters apart, different objects. Is this concept clear about limit of resolution? Any doubt? Any doubt in this part? Okay, okay, so what you understand is in microscope, let's say there are two people, okay, from the batch, um, okay, very long names, no problem. Let's say there are two people, or you can say, yeah. So what happens is if one is A, another is B, and if the minimum distance between you is something that is 1 meters, then I can perceive A and B as different objects, okay. That is something that is so what is the reason for the limit? So this limit of resolution, this is the limit of the magnifying power of your, the power of your lens that is there. So for example, our eyes, the lens of the eye, so that can visualize two objects if they are far apart by 1 meter, okay, minimum distance should be 1 meter. If it is more than 1 meter, then yes, you can just say that A and B are separate entities. But if the minimum distance is lesser than 1 meter, then what happens is you perceive them as a single person, okay. You don't know whether it is A or B, you just perceive them as a single person, because the distance now is, let's say, 0.5 meters, so then you can't distinguish between them. Is this clear? No, it is not used to capture two objects, like to observe. Like let's say you are seeing two cells, okay. So let's say these are two cells. If the limit of resolution of your microscope, let's say this is micrometers. So if it is 1 micrometer, but the cells are 10 micrometers apart, you will see the two cells as separate entities inside when you look into the microscope. But if let's say the cells are very close, right? So let's say they are 0.5 micrometers, then you will see a single cell. You can't distinguish it. That is something. Is it clear? Okay, clear. Okay, good. So this was about limit of resolution, and you will study this in physics also. The main thing is the formula for limit of resolution, that is 0.61 lambda divided by n sine theta. So this is the formula for limit of resolution, okay. So in this formula, lambda is your wavelength, right? Lambda is your wavelength, n is the refractive index, okay, refractive index between the lens and specimen media. So in this image, if you see here, you will put your specimen, right? This is the lens, this is the objective lens. So in between the specimen and the objective, this medium, this is air, okay, and the refractive index for air is 1. So that is something, that is the n in this formula, okay. So if you see, yeah, I hope this is clear. So if you have this objective lens, this is the specimen, the media between the two, that is the air, has a refractive index of 1. So that is how that is the n in the formula. And now what is sine theta? So sine theta is nothing but, if you see here, what happens is, so the light that is coming, okay, so if you see, so you see this case, right? So the light that is falling into the lens, right? So you can imagine it as a cone of light, right, as a cone of light. So now in this particular case, this half angle of the cone, that we can call as theta, right, that we can call as theta. So this half angle is theta, correct, and this is the theta that I am talking about in this formula, the half of the angle subtended by the cone of light, okay. So is it correct that the microscope with lesser resolution is considered? Yeah, I am coming over to that point, but first understand what is the theta. I hope you understood it. The half of the angle of the cone, that is we call as the theta, right. So if you talk about this theta, so what happens is, now there is a very important relationship that you should understand, that the limit of resolution is inversely proportional to the resolving power, okay. Limit of resolution is inversely proportional to resolving power. This is resolving power, right, this is resolving power. So how we are saying this, right? So I will make you understand this thing, okay, in a simple way. So let's say there are two microscopes, one is A, another one is B, right? So for this microscope, I am saying that the limit of resolution value that is there, okay, let's say this is 1 meter. Then for another, the limit of resolution is 0.5 meter. So you guys comment down that which is a better microscope, one which has a limit of resolution as 1 meter, or the one which has a limit of resolution as 0.5 meters. So if glycerine or any dye is poured, should we take the refractive index of that medium? Correct, that refractive index will be taken. So many people are saying B is a better microscope. Absolutely correct. B is a better microscope. Why? Because the limit of resolution is less. Now why? Because it can distinguish objects which are even 0.5 meters apart. Are you understanding this point? It can distinguish objects which are even 0.5 meters apart. But if the objects are, let's say, you can say, for the A microscope, let's say the objects, they are 0.75 meters apart. So this microscope will not be able to detect these two objects as separate cells or separate entities. But this microscope, right, if you consider, the minimum distance required is 0.5 meters. But if the distance is 0.75, then it can distinguish them as separate entities. So is this part clear? Why B is a better microscope than A? So we say that the resolving power of B is greater than A because the limit of resolution of B is less than A. Is this relationship clear? Okay. Okay, good. Okay, nice. Yeah. So this is clear to everyone. Good. So if this is clear, then this is something. So what happens is, as you bring the microscope closer to the specimen, correct? So this was the initial position, then you are bringing it closer, then you are bringing it closer. So what is happening is, this cone of light, the angle is increasing, right? Initially the angle was this, now it is increasing, correct? This theta is increasing as you are bringing this lens closer to the specimen. Now if you know about one thing, that let's say sine 0 is 0, sine 30.5, sine 90 1, correct? So as you increase the theta, sine theta value increases. Everyone agrees on this point. As you increase the value of theta, sine theta value increases, correct? So in this formula that I wrote, that is d is equal to 0.61 lambda divided by n sine theta. In this formula, if sine theta increases, limit of resolution decreases, correct? If sine theta increases, limit of resolution decreases, right? So that means the resolving power increases. That is better for us. So that's why you have observed, whenever you see in the microscope, if you are at this much distance, then you will not observe the cells clearly. You will have a very haphazard kind of a image, right? A very blur image. But as you bring this close, right? As you bring this lens close, you observe that you are observing the image much clear, right? That is why, because the sine theta increases, limit of resolution decreases and resolving power increases. Clear? Very good. So now what you have to understand is, what could be the maximum value of sine theta for this particular thing? What could be the maximum value of sine theta? So maximum value could be sine 90, that is 1, right? If you put theta as 90 degree, then you can get the limit of resolution value, okay? That could be, if this is 1, then this part could be minimum and resolving power can be maximum, correct? Now, but 90 degree is not practically possible, because if it is 90 now, what happens is, if this is so close, then this will just bombard with the sample. So that is not possible in practical purposes. So for practical purposes, what happens is, this theta value, which is approximately 74 degrees, right? At this particular value, this limit of resolution, this is minimum, and the resolving power is maximum. So that is something that happens in practical purposes. Maximum you can go to this angle, that is around 74 degrees, not actually 90 degree, because then this will just crash with the specimen. Okay, is it clear? Yeah, this is relevant. I will show a PYQ. Okay, but the thing is, based on this concept, you can get different questions in NEST. Okay. Yeah, so if now you can see, this is direct and inverse relationship. So if you decrease the wavelength, then also the limit of resolution will decrease, because it is in the numerator. If you change the refractive index, if you increase the refractive index, then also the limit of resolution will decrease. Okay. Yeah, around 74 degree, that is the max angle that you can go, right? As sine 0 increases, the denominator will decrease, so d will, no, as the denominator, see, as, yeah, if you are taking it as a sine 0, then this is less, so this d will increase, that is correct. Yeah. So now what happens is, this was about the different factors. So if you decrease wavelength, you decrease the limit of resolution. If you increase the theta maximum till 74, you again decrease the limit of resolution. And now comes the refractive index. So I hope you have heard about immersion oil, right? That we put sometimes to see even at much more magnification, correct? So if we talk about the immersion oil, what happens is, when you are putting oil here, right? So oil, it has a refractive index of 1.5, and that immersion oil, and this glass that is there, the glass refractive index is also 1.5. So all the light rays which are coming, they are not bending, right? They are not going away from normal, and all the light rays are falling to the lens. So that is the idea that we put immersion oil, right? Because if you put immersion oil, you change the refractive index from 1, that was for air, to the 1.5, that is for the immersion oil, and the glass refractive index is also 1.5. So all the light that is falling to the lens, that is not bending, because you know that when light moves from rarer to denser, it bends, right? And that is how these things are important. Now we have a question. So on microscope, This was asked in NEST 2016. Okay. So the question is, students in the biolab were asked to find the size of a single cell. They were given a compound microscope with an eyepiece of 15x magnification and an objective lens of 10x magnification. The eyepiece has a scale of least count of 10 micrometers. When students focus under the microscope, they found that one cell measures 60 divisions of eyepiece scales. What is the approximate size of the cell? So can you guys try this question? So immersion oil is a kind of oil that is commercially available for the microscopes. If you put that oil, that has a refractive index exactly as 1.5. But if you don't have that much money, you can put coconut oil, but that will have a refractive index which is lesser than 1.5, that is around 1.42 or so. So you will lose some light, but that is still like manageable. So try to solve this question. This is based on that formula only, that the net magnification is the product of the objective and the eyepiece magnification. But the twist here is that for the eyepiece lens, here you are given the least count. So what is least count? Least count, okay, why the oil is used? Okay, so see, what happens is, let's say, here you are having the lens, correct? This is the stage, this is the slide, and this is the sample, right? And you are getting the light rays, correct? So yeah, so you are getting the light rays here, correct? And now what happens is, this refractive index of the media before putting the immersion oil, that is 1, that is for air, right? Now you put the immersion oil. So once you put the immersion oil, what happens is this refractive index becomes 1.5. Now we compare the two cases. So let's say this is case A, this is case B. For case A, the limit of resolution formula is 0.61 lambda divided by the refractive index value is 1, right? And sine theta. For this case, this value is 0.61 lambda divided by 1.5 into sine theta. So now you can see here the denominator, you can see in this case the denominator value is more. So this fraction becomes lesser. That means the limit of resolution value is lesser for B. That means the resolving power for this particular microscope with the immersion oil, that is higher. So that means that the dB, because dB is less than dA, then we can say that the resolving power of the second case, that is higher as compared to the resolving power in the first case. So I hope this is clear, right? This is clear to everyone. So why we are using the oil? Because if we use the oil, we are decreasing the limit of resolution, increasing the resolving power, right? Now you need to solve this question. So what is least count? Least count is the minimum value that you can measure for any of the instrument, correct? So that is something you called as a least count. So here the least count of this is 10 micrometers. So if you have to visualize this, what you can see is, let's say you are having a scale. So this is 0 centimeter, this is 1 centimeter, and here you are having 10 divisions, right? So this smallest value that you can measure, this smallest value, this will be what? This will be, if you see, this whole is 1 centimeter divided by 10 divisions. So each division is nothing but it is 0.1, correct? That is 0.1 centimeters. So this is each division. So this is the least count for this scale. Similarly, for the eyepiece, if the least count value here, this is, okay, if this value here, it is 10 micrometers, and here you are having how many divisions? So one cell measures 60 divisions, right? So here you are having 60 divisions. So now if you have to calculate the value for the size of the cell, right? For the size of the cell. that is not the actual size. This we can call as the apparent size, as visualized by the eyepiece, because eyepiece is magnifying the image, so this is not the actual size, correct? So if you want to calculate this apparent size of the cell, that will be least count into 60. That will be 60 into 10, right? That will be 600 micrometers. So this is the apparent size. Now what is the actual size, right? So to calculate the actual size, what you need to understand is, let's say the actual size, if we are writing, we are multiplying the actual size with the magnification of the microscope, that is the overall magnification of the microscope. Then we are getting something that we called as the apparent size of the cell. Apparent size of the cell. So this is something that is a logical formula, not something that you have to learn. Simply what is the actual size, that will be magnified sometimes and that will become the apparent size, correct? So that is how this actual size is being magnified. Now if you understand, what happens is, you have to calculate the actual size, right? So what we will do is, let's say this quantity is x, right? What is the overall magnification? So overall magnification is MO into ME. MO is for objective, ME is for eyepiece. So the eyepiece is 15x, objective is 10x. So 10 into 15, that is 150. This is the overall magnification of this microscope. Now in this formula, if you put 150 here, and the apparent size that I calculated based on the observation, that was 600. So with this you get x as 600 divided by 150, that is nothing but 4. So your answer is 4 micrometers. That is nothing but the actual size of that particular cell, and that is the option number C, right? Why least count into number of division gives the apparent size? So see, what happens is, in this particular case, so whenever you are observing something, right, whenever you are observing something, so what is happening is, this is the size that you get from the observation, that is a magnified version. So let's say this person Uday, okay, so let's say Uday, you are some 150 centimeter tall, right? This is your actual size. Now I magnified and I am able to see you. I am saying that you are 1500 centimeters tall. So this is something that I am able to observe because of the microscope, because you were magnified 10 times, right? So now what happens is, this thing, this 1500 centimeter, this is your apparent size, not your actual size. And this 150 centimeters, this is your actual size. So what happens is, if you multiply the actual size with the magnification value, you get the apparent size. That's how we calculated this thing here. Is it clear? Yeah, option number C is correct. The question says, what is the approximate? Yeah, the approximate size, because whatever you are calculating in this particular sense, that will have some error, right? So that is why it is saying approximate, but approximate and apparent, they are different. Okay, I hope this is clear. Okay, clear. Okay, good. So I think we don't have much time to start the next topic, that is hemocytometer. But what we will do is, we will see a basic thing that is required for this, that is dilutions. Okay, how to make dilutions. So this is something that will be helpful in a lot of your nest numericals, and also it will be helpful if you are working in lab, because this is something that you will have to do every day. Okay, so we will cover this, we will end the class with this, and then we will start hemocytometer in the next class. Okay, so if we talk about dilutions, let's say in this test tube, right, let's say you are having some reagent, okay, any sort of reagent you are having, or let's say you are having any particular thing, let's say you are having coffee here, okay, and you have to dilute this coffee, right? So what you did is, you took, let's say 1 ml of coffee, right, and you added 9 ml of water, correct? So once you do that, what happens is you have diluted this 10 times, because now the overall volume in this test tube initially, let's say from this was some value, right? Let's say this was 5 ml, you took 1 ml out and you put 9 ml water, so this becomes 10 ml. So this is the final volume, right? So what we can say is that this was diluted 10 times, right? This was diluted 10 times. So this we call as the dilution factor. If you write over here, that will be 10, because this what is dilution factor? So if you understand this you can say as the final volume divided by the initial volume that you are having. So let's say this was 10 ml finally, initially it was 1 ml, so that becomes 10. So this was diluted 10 times. This is how you understand dilution. So is this part clear to everyone? Any doubt here? Is it clear? Okay, good. So since it is clear, now what we will understand is, let's say you have to make different dilutions, right? So let's say you have 100 mg of, let's say, coffee powder, right? And you put this in 100 ml of water, right? So this is 100 mg of powder in 100 ml of water, right? So what this actually becomes is 1 mg per ml. This is the concentration. This is something we call as concentration, that in per ml of the solution you are having 1 milligram, right? So this is the concentration. And now if let's say I am diluting it 10 times, 10 times I am diluting it, okay, then what will happen is this 1 mg will reduce to 0.1 mg per ml of concentration. Let's say I am again diluting it 10 times, right? Then it becomes 0.01 mg, like this. So like this you keep on diluting the thing and you will decrease the concentration, right? So to do this all in lab, so let's say you have this formula that you call as C1V1 equal to C2V2, where this part represents the stock solution. Stock solution means this part. This 1 milligram is 10 to the power minus 3 grams. No, you can't write it as 1 mg per 10 ml, because you have 100 mg in 100 ml, right? So if you cancel this, you get 1 mg per ml, right? So that is how, that is the concentration you are getting. And the thing is that this is the stock concentration, because this is larger, right? It is larger in concentration, 1 mg. But if you talk about 0.01 mg, this is smaller. So for this part we call it as a working, for the working solutions. That is the one which will be used for the experiments, right? So let's say if you want to go from this particular concentration to this concentration, you need to find how to do that. Then let's say in place of C1 you are putting 1 mg per ml, right? And you need to know how much of this is required to make this concentration, that is 0.01 mg per ml, right? So this is 0.01 mg per ml concentration, right? And let's say we want to make some 10 mls of it, right? So how much of the volume of the stock is required in which you will add water, right? So that is how you calculate it as x is equal to 0.01 into 10, that is 0.1, right? You will get this as x is equal to 0.1, right? And this will be ml. So if you calculate this, this is the thing. So that is how you can find that what volume of the stock is required, and if you subtract this 10 with 0.1, the remaining water you can add to calculate this working, the volume of the stock that is required to make the working concentration. Okay, so does concentration have any relation to density? So if your sample, if solution is more concentrated, then the density will be more. If it is diluted, then it will be less. and this is how you calculate the dilutions, and we will use this for the hemocytometer, right? So let's say we have, so if I tell you one thing, let's say here you are having 100 cells, right? And you add in this solution, right? And you add here 400 ml of water. So 100 cells, here you are having 100 ml of cells, right? And you added 400 ml of water. So now you can tell me that the final this thing that will be produced, this is how much times you are diluting the cells, how much times you diluted the cells. Can you answer this? How much times did you diluted the cells? If initially you have 100 ml of cells, you added 400 ml of water, right? So everyone is saying we diluted 5 times. Correct. Because I told you what is dilution factor. This is the final volume, that will be 400 plus 100, that will be 500. And the initial volume is 100. So you can say you diluted it 5 times. So that is how you carry out these dilutions, right? And one more thing that we can say is, let's say you have to carry out 1:10 dilution, right? For any particular solution, you have to make a 1:10 dilution. Then how you can do this? You have to think it in this way. You have to subtract 1 from this, right? So you can say you have to make in 1:9. So this will be your sample, right? And this will be your water. Now, if you multiply it with any number, let's say you multiply it with 10. So, do you have 10 microliter of cells? Okay. And if you add 90 microliter of water, then what happens is this, if you understand, finally it will become 100. So, 100 upon 10, this is 10 times diluted. The dilution factor is 10. Now, let's say if you multiply this with 20, if you want to take more volume, then 20 microliter of cells plus you have in this case 180 microliter of water, then the final volume will be 200 divided by 20, again it is 10 times diluted. So the dilution factor is 10. Is this part clear to everyone? How you can make different dilutions? So let's say you had one solution and I asked you to dilute it 10 times, right? So if you have to dilute this 10 times, then if I write it in this way, dilute the solution 10 times, right? So this is how you have to, this flow you should understand, that you can just simply carry out this approach and you can arrive at the answer. Is it clear to everyone? Any doubt? So I hope this is clear, and in the next class we will start with hemocytometer. That is a technique you use to count the cells. So this we will cover in the next class, and we will solve the PYQs also. So practice questions as well as the next PYQ, right? So okay, I hope this was clear, and thanks a lot for joining the class. And this is the QR code that you can scan to go to the, to be redirected to the feedback form, where you can write like how was the class and any suggestions, right? And all those things. So yeah, do you have any other doubts? Okay, great. So let's meet in the next lecture where we will cover the remaining topics. Okay, thanks a lot for joining. Yeah, bye. कितना टाइम हुआ? जल्दी हो गया होगा।